DNA fingerprinting or DNA profiling. Okay, it is very important topic. Okay, DNA fingerprinting is the present day genetic detective in the practice of modern medical forensics. The structure of each person's genome is unique. The only exception being monozygotic identical twins. Okay. Monozygotic identical twins means twins developed from a single fertilized ovum. Okay. Twins developed from a single fertilized ovum. Okay. That is called monozygotic identical twins. The unique nature of genome structure provides a good opportunity for the specific identification of an individual. Okay. It may be remembered here that in the traditional fingerprinting technique, the individual is identified by preparing an ink impression of the skin folds at the tip of the person's finger. This is based on the fact that the nature of these skin folds is genetically determined, and thus the fingerprint is unique for an individual. Okay. In contrast, the DNA fingerprint is an analysis of the nitrogenous base sequence in the DNA of an individual. Okay. It is very important. The DNA fingerprint is an analysis of the nitrogenous base sequence in the DNA of an individual. Okay. The original DNA fingerprinting technique was developed by Alec Jeffries in 1985. Okay. Next. Although the DNA fingerprinting is commonly used, a more general term, DNA profiling, is preferred. Okay. Next, what is the definition of DNA fingerprinting? The DNA fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a criminal investigation. A DNA sample taken from a crime scene in compared with a DNA sample from a suspect. Okay. Next, the steps of DNA fingerprinting. Okay. The DNA fingerprinting involves a number of intensive and important steps in order to fully complete and develop an DNA fingerprint of a father, a suspect, or person involved in an immigration problem. Okay, first step, look at here. The process of DNA fingerprinting starts with isolating DNA from any part of the body, such as blood, semen, vaginal fluids, hair roots, teeth, bones, okay, etc. Look at here. First, you take blood sample, okay. This is example, okay, blood sample. First, you take blood sample. Next, the DNA extracted from blood cells. Okay. This is DNA. Look at here. This is blood sample. First step, you take blood sample or semen, vaginal fluids, hair roots, teeth, bones, etc. Okay. Next step, polymerase chain reaction, PCR, is the next step in the process. In many situations, there is only a small amount of DNA available for DNA fingerprinting. Because of this, in a test tube, the DNA replication is must occur to make more DNA. The DNA and the cells will undergo DNA replication in order to make more DNA to be tested. Okay. After the DNA is isolated and more copies of the DNA have been made, the DNA will be tested. The scientists will treat DNA with restriction enzyme. The enzymes cut the DNA near specific recognition nucleotide fragments. Look at here. Next step, DNA is cut into fragment by restriction enzyme. First step, you take blood sample. Next step, DNA is extracted from blood cells. Okay, this is the DNA. Next step, DNA is cut into the fragments by restriction enzyme. It can be used restriction enzyme. DNA will be cut the DNA fragments. Okay. Next step, agarose gel electrophoresis. Okay. Okay. This will, okay. The DNA is cut into the fragment by restriction enzyme. Okay. This will produce different sized fragment which are known as restriction fragment length polymorphism, RFLPS. It can be used. Okay. Cut the DNA fragments. It can be used RFLP. These fragments can then be observed doing an experiment called gel electrophoresis, which separated DNA based on fragment sizes. Okay. What is meant by restriction fragment length polymorphism? Okay. A RFLP represents a stretch of DNA that serves as a marker for mapping a specified genes. RFLPs are located randomly throughout a person's chromosomes and have no operant function. Okay, DNA molecule can be cut into different fragments by a group of enzymes called restriction endonucleases. These fragments are called polymorphisms. Okay, that is called restriction fragment length polymorphisms. Okay, next step, these fragments. Okay, these fragments means DNA fragments. Okay, these fragments can be observed during an experiment called gel electrophoresis, which separated DNA based on fragment sizes. Okay, look at here. Look at here. This is the agarose gel electrophoresis. First, you take blood sample. Next, this is the DNA. Next, it can be used RFLP. Okay, restriction fragment length polymorphism. Next, DNA is cut into the fragments by restriction enzymes. Cut the DNA fragments. Okay. Next step, agarose gel electrophoresis technique. Okay. The DNA fragments are separated into bands during electrophoresis in an agarose gel. Okay. Look at here. Gel electrophoresis is the next step in this process of DNA fingerprinting. During gel electrophoresis, an electrical current is applied to a gel mixture, which includes the samples of the DNA. Okay, samples of the DNA means blood samples of the DNA. Okay, next, the electric current causes the DNA strands to move through the gel. This separates the molecules of different sizes. The fragments of separated DNA are sieved out of the gel using a nylon membrane. Okay, treated with chemicals that allow for it to break the hydrogen bonds of DNA, so there are single strands. Okay, look at here. This is spelling mistake. Look at here. This is agarose gel electrophoresis current is supply. The DNA fragments will be moved. Okay. Next, after agarose gel electrophoresis completed, these gel transfer into the nylon membrane. Look at here. The DNA bands pattern in the gel is transferred to a nylon membrane. Look at here. This is nylon membrane by technique known as Southern blotting technique. Already I told, okay, Southern blotting technique. Next, okay, Southern blotting technique means detection of detection of DNA molecules, that is called Southern blotting technique, identification of DNA molecules. Next step, okay, the radioactive DNA probes is prepared. Okay, this is radioactive DNA probes. These probes bind with the specific DNA sequence, okay, of the on the membrane. Okay, what is mean by probe? Probe is prepared. Okay, probe means a labeled molecule used in hybridization technique. Okay, this is the DNA probes. These DNA probes bind to specific DNA sequences. Okay, okay, how do you understand? These DNA probes binding with the DNA fragments. Okay. Next step, washing. Excess DNA probes is washed off. Okay. This is radioactivity probe is bound to the DNA pattern on the membrane. Okay. Next step, X-ray, UV light. Okay, the DNA single stranded is cross-linked against the nylon using heat or UV light. Okay, the DNA fragments binding with the nylon membrane. Okay, it can be used UV light. Okay, exposed into the UV light. Next step, the probe. Okay, are you understand? The probe shows up on photographic film because the strands on DNA and give off light in the end it leaves dark spots on the film, which are also known as the DNA bands of a person. Okay, what make up the fingerprint or the unique pattern of bands? The pattern of bands are different because we are all different and unique other than identical twins. Okay. Look at here. Okay, this is the DNA pattern. Okay. Next step, X-ray film is placed next to the membrane to detect the radioactive pattern. Okay. Next, the X-ray film is developed to make visible the pattern of bands which is known as the DNA fingerprint. Okay, look at here. This is the DNA fragments. Okay. Okay. Next, X-ray film to develop auto radiograph. Okay. This reveals specific bands. Okay. This is the DNA bands, the DNA fragments. Okay. This is the DNA fingerprint. Okay. Are you understand? This is steps of DNA fingerprinting or DNA profiling. Okay. First, you take blood sample. Next step, DNA is extracted from the blood sample, blood cells. Okay. Next, it can be used RFLP, resistant fragment length polymorphism technique. It can be used. Okay. This is the DNA. DNA is cut into fragments by restriction enzymes. Okay. It can be used restriction enzymes cut the DNA fragments. Next step, agarose gel electrophoresis technique. Okay. The DNA fragments are separated into bands during electrophoresis in an agarose gel. Okay. The DNA bands pattern in the gel is transferred to a nylon membrane. Okay. It can be exposed to the UV radiation. Okay. The bands, the DNA bands bind with the nylon membrane. Okay. It can be used Southern blotting technique. Next step, the DNA probes, radioactive DNA probes is prepared. Okay, these probes binding with the DNA sequences. Okay, specific DNA sequences. Next, wash. Okay, excess DNA probe is washed off. Some DNA probes binding with the DNA fragments. Okay, this is ninth step. The radioactivity. This is, look at here. This radioactive DNA probes binding with the DNA fragments. Okay. Next step, X-ray film is placed next to the membrane to detect the radioactive pattern. Okay. Next step, X-ray film to develop autoradiograph. This reveals specific bands. Okay. The X-ray film is developed to the make visible the pattern of bands, which is known as DNA fingerprint. Okay. Look at here. This is just a protocol. Look at here. First, you take DNA sample. Next, extraction of DNA. Okay. This is the DNA. Look at here. Next step, restriction enzymes. It can be used, cleave the DNA by restriction enzymes. This is the DNA fragments. Next step, gel electrophoresis technique. It can be used to separate DNA fragments. Look at here. This is the DNA fragments. Next step, Southern blotting technique. Okay, this is the stock. Okay, this is the gel. This is nylon membrane. Okay, these DNA fragments binding with the nylon membrane. Okay, this is the sandwich preparation. Okay, it can be used UV radiation. Okay, next autoradiography to obtain DNA bands. Okay, DNA probes binding with the DNA fragments. Okay, next developed and visualized DNA fragments. Okay, this is UV, uh, sorry, X-ray film to develop autoradiograph. Okay, it can be reveals the specific bands, DNA bands. Okay, this is the DNA fingerprints. Okay, any doubts? This is the steps of DNA fingerprinting. Okay, first scientist Sir Alec Jeffreys was okay. DNA fingerprinting technique was developed by Sir Alec Jeffreys. Okay, next DNA fingerprinting definition. DNA fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a criminal investigation. Okay, a DNA sample taken from a crime scene is compared with a DNA sample from a suspect. Okay, next steps. This is a diagram. Okay, it is very, very important. Next, DNA fingerprinting steps. Okay, are you understand? This is the protocol. Okay, any doubts? Okay. Look at here. First step, the process of DNA fingerprinting starts with isolating DNA from any part of the body, such as blood, semen, vaginal fluids, hair roots, teeth, bones, etc. Okay, next step, PCR, polymerase chain reaction. PCR means polymerase chain reaction. Okay, the double stranded DNA of interest is denatured to separate into two individual strands. Each strand is then allowed to hybridize with a primer. Okay, that is called renaturation. The primer template duplex is used for DNA synthesis. The enzyme DNA polymerase. Okay, the PCR technique in 3 steps is there. Okay, first step denaturation, renaturation, and synthesis. Okay, that is called PCR. Next, there is only small amount of DNA available for DNA fingerprinting. Because of this, in a test tube, DNA replication is must occur to make more DNA. Okay. Next, the DNA and these cells will undergo DNA replication in order to make more DNA to be tested. Okay. DNA is isolated and more copies of DNA have been made, the DNA will be tested. The scientists will treat DNA with restriction enzymes. Okay, the enzyme that cuts DNA near specific recognition nucleotide sequences. Nucleotide sequence means adenine, guanine, cytosine, thymine. Okay, that is called nucleotide sequences. Okay. Next, restriction fragment length Next step, RFLPS, it can be used. Restriction fragment length polymorphism. Okay, these fragments can then be observed doing an experiment called gel electrophoresis technique. Next step, gel electrophoresis technique. Okay. Gel electrophoresis is the next step in this process of DNA fingerprinting. During gel electrophoresis, an electrical current is applied to a gel mixture, which include the samples of the DNA. Look at here. This is the agarose gel electrophoresis. The samples load into the wells. Okay. Are you understand? The DNA migration towards migration of DNA is dependent on the size and shape. Okay. In general, small linear fragments move faster than larger DNA fragments. Okay. This is called gel electrophoresis technique. Next step, Southern blotting technique. Okay. The DNA is cross-linked against the nylon membrane. It can be used. Okay. UV exposing into the nylon membrane. Okay. The single-stranded DNA binding with the nylon membrane. Okay. Next, probes, radioactive probes binding with the DNA strands. Okay. Give off in the light and the leave dark spots in the film, which are also known as the DNA bands of a person. What make up the fingerprint or the unique pattern of bands? The pattern of bands are different because we are all different and unique. Okay. Okay. Are you understand? This is the DNA fragment. Okay. Any doubts? DNA fingerprinting steps or DNA profiling steps. Okay. Are you understand? Any doubts?